Hello and welcome to today's webinar, which is focused on ensuring accurate classification of variants in BRCA1, BRCA2 and HRR genes. Next slide, please. We welcome you very much, along with myself, Sandy Deans, who's the Director of GenQA, and my colleague Simon Patton, who's the CEO of EMQM. We've got a couple of housekeeping rules just to start before we start and hand on with the presentations from our experts today. Your microphone has been automatically muted just because there's many people on the call and to avoid background noise. We've also recorded this webinar for purposes for you to be able to review and listen to it after the event. And also, this is very much an interactive session. It's educational, but we also want some input and to actually respond to your questions. So please use the Q&A function to submit your questions on the Zoom call, and we will answer them hopefully at the end of the dedicated Q&A session, which follows our expert presentations today. Next slide, please. Okay, as I mentioned, these really are educational webinars, a series that we're putting together in 2022, and we want to make sure that they're actually fit for purpose and actually fulfill your needs. So your feedback is incredibly important to us, and it does help us to continue to provide this high quality training to the healthcare professionals globally. So you'll receive an email after this masterclass to a link to a short feedback survey, and we would really appreciate if you could just take a couple of minutes of your time to complete the survey. We do review the feedback and adapt the content of the webinars accordingly. Next slide, please. So let's kick off with today's webinar. As I mentioned, myself and Simon will be chairing, and we've joined today by three members of our expert panel who are heavily involved in delivering the external quality assessments for classification of variants. And also today we are going to provide some educational feedback on using databases, applying the guidelines, and really starting to help you how you can implement classification in a standardized form in your own clinical service. So next slide, please. Quick disclaimer. Thank you. And moving on again. Okay, so we have three expert reviewers today to join us. We start with Dr. Andrew Morani, who is based in Edmonton in Canada, so has a very early start to join us on this live webinar. He works within the Alberta Health Services and is a clinical molecular geneticist with extensive experience in the interpretation of cancer variants, and he joined the EQA team last year. Andrew is going to give us a general overview of the variant classification guidelines that are currently exist. Then we move on to Miranda Durkee. So Miranda is a lead clinical scientist for rare diseases at Sheffield Diagnostic Genetics Service here in the UK. And Miranda's really been instrumental in the CAN-VIG work and is author of also the 2022 guidelines. So is an expert person to come and talk to us today around the classification codes, how to use them and when to use them, and give us some insight into the actual application of the guidelines. And then finally, we have Professor Kathleen Claus from Ghent in Belgium. Kathleen has a wealth of experience in cancer predisposition and precision oncology, and again has heavily involved in the Enigma working groups on the international stage. So again, very well placed to help today give us a great overview of this ability to accurately classify the variants. So I'm now going to move on. So next slide, please, and hand you over to the safe hands of Andrew, who is going to start us off with our presentations. Andrew, over to you, please.
Thank you for that introduction. There we go. All right, so this slide just shows the different guidelines that people are using for these schemes. So the options we have are the ACMG 2015 only, the ACMG 2015 and ACGS 2020, or the ACMG 2015, ACGS 2020, and CAN-VIG Okay. Almost half of the individuals registered for the scheme use the ACMG 2015 only, and then the remainder use one of the other combinations, including other. So in 2015, the ACMG and AMP published the standards and guidelines for variant interpretation, and this set the foundation for standardizing variant interpretation across not only the country, but I guess across the world for clinical labs. I apologize, I'm reading that the sound is not great. Please let me know if this is getting better. So after the 2015 guidelines were published, there were a number of other consensus statements and guidelines that came out from groups such as the ACGS and the BCGA. There are several best practice guidelines that came out, as well as from the CanVig UK group. So some considerations to keep in mind when reviewing variant interpretation. The first is the term mutation and polymorphism. Because they are somewhat ambiguous, they should not be used and have been replaced with the term variant. Variant can be classified into 5 different categories: pathogenic, likely pathogenic, uncertain significance, likely benign, and benign. These recommendations are intended to determine whether a variant in a gene that's already has a definitive role in Mendelian disorder, whether this variant may be pathogenic for that particular disorder. Pathogenicity determination should be independent of interpreting the cause of disease in a given patient. Which means an example of this is if you have a large panel that you're looking at, it is possible to find a pathogenic variant in an individual that does not necessarily explain the disease for that individual. The mechanism of disease is important to consider when evaluating a variant. Not all variants, not all genes are loss of function. Some are gain of function genes, such as rosopathies. These guidelines do not work great for somatic and mitochondrial variants, which have some special additional considerations to keep in mind. And variants of uncertain significance should not be used for clinical decision making. One point I did not mention here is these guidelines work best for highly penetrant disorders. When the penetrance decreases, a lot of the evidence you're evaluating for variant classification becomes a lot more tricky to interpret. Next slide, please. I don't see. Thank you. Variant classification information can be broken down into several different categories of evidence, such as population data, computational predictive data, functional data, segregation data, and allelic data. The majority of these classes of evidence have both benign and pathogenic leading evidences, with the exception of de novo data. De novo only has evidence that leads towards pathogenicity, not to benign. Next slide, please. So this is a busy slide. It lists all the evidence in the 2015 Richard et al. Publication for variant classification. But what I just wanted to point out with this is the evidence can be broken down into, on the pathogenicity side, very strong, moderate, and supporting. And on the benign side, standalone, strong, and supporting. The other thing I wanted to point out is you can probably notice from the lists of these tables that there are more pieces of evidence that can be used to point towards pathogenicity than there is towards benign. Next slide, please. So final variant classification. So final classification of a variant. Once you've identified all the evidence you can for a variant, what you do is add up the different points of evidence and the strengths, and then you determine whether it adds up to being a pathogenic change, a likely pathogenic change, benign, or likely benign, or of uncertain significance. Next slide, please. So this was a great start for variant classification and gave a lot of information, but after the publication of this, there are several other publications and guidelines that came out to help give more information for this. And one such publication is the modeling of these ACMG guidelines into a Bayesian classification framework, which came out in 2018. And what essentially they did is they took the strengths of evidence from the 2015 guidelines and followed a Bayesian model to try to quantify the evidence used. And what they were able to do is with this, find a pattern that fit the evidence, and also came up with 3 new variant classification combinations that led to likely pathogenic, and uncertain significant classifications. Next slide, please. Another important publication that came out subsequent to the 2015 guidelines is a in-depth review of the PVS1 variant criterion. And what this publication did is they looked at the PVS1 evidence and they broke it down into 5 different categories. And I'm not going to go through the whole decision tree that's on the left here, but work you through one example from a previous scheme. And so from this scheme that happened a couple cycles ago, it was a BRCA1 5561 del. This is a frameshift variant, so it would follow the first scheme, first point in the decision tree. It's a non-shifter frame. It's a non-sensitive frameshift. This variant occurs in the last exon, so it's not predicted to undergo nonsense-mediated decay. The functional role of this protein region is unknown, and less than 10% of the protein is affected. So we end up with a PVS1 moderate strength. Next slide, please. Another publication that came out is a deep dive into the BS3 criterion. So this is the use of functional evidence for classification of a variant. And this publication goes through how to evaluate the relevance of a particular assay for your gene or variant of interest, how to evaluate whether the assay was performed appropriately with the correct number of controls, and ultimately how to apply the evidence for your particular variant of interest. Next slide, please. Since the 2015 guidelines, there have been a number of other updated sequence variant interpretation recommendations. Not all of them were published in the literature, but they all can be found on the ClinGen website. Next slide, please. One of such is the ClinGen recommendations for PM2. It was recommended that PM2 be downgraded from a moderate strength of evidence to a supporting strength of evidence, partly because it was found that in the exact database, over 54% of high quality variants were only seen once in the entire data set. So this suggests that being a rare variant is not a rare occurrence, and therefore the strength of this evidence should not be so high. However, by decreasing the weight of this criterion, they also recommended the change of allowing PVS1 and 1 supporting strength of evidence to also be considered likely benign. Next slide, please. Another such recommendation is how to use the in-trans criteria, or PM3. Considerations to use for this include allele frequency, phasing, classification, and homozygous occurrences. And essentially what you do with this is there's a scoring criteria based on the strength of the in-trans pathogenic, the in-trans variant you're seeing the variant with, how many times it occurs, and whether it's confirmed phase or not. And then you add up your points, and then there's a second table where you can figure out which strength of evidence you should use for this. Next slide, please. There are also several ClinGen gene or disorder specific guidelines that were created by separate working groups, which can also be found on the ClinGen website. Next slide, please. One of such is a fairly recent one for the ACMG. And what's useful about these guidelines is they are based on the 2015 ACMG guidelines, but they're refined for the gene of interest. So for ATM, they've established better BA1 and BS1 criteria, as well as which rules do not apply for this gene of interest, such as BS2. And so they're very useful in taking out the guessing work for applying the ACMG guidelines to the ATM gene, or whichever gene of interest the guidelines are made for. Next slide, please. So at the same time that the CanVig, the Cancer Variant Interpretation Group UK, worked on their own set of guidelines, which is modeled off the ACMG guidelines. However, these guidelines, as they're set for cancer genes, they did give a little more detail than the general 2015 ACMG guidelines. Next slide, please. These guidelines are very useful because they took out a lot of the guessing work, as did the ClinGen gene-specific guidelines, but they established exactly what would constitute for, so for this example, for PM2, how to score when to use PM2. So whether it's 0 or 1 alleles in greater than 50,000 individuals. So they took out a lot of the ambiguity in some of the ACMG guideline recommendations. Next slide, please. CanVig UK also has several gene-specific guidelines, such as for SDHB, for MMR genes, and for BRCA1 and 2. Next slide, please. These recommendations are also very useful because, like the ATM guidelines from ClinVar, these spell out exactly which criteria can and cannot be used for the BRCA1 and 2 genes, and what the thresholds that you should be using are, and even which functional evidence is useful for the classification of these variants. I'm also told that Enigma and ACMG will be releasing their own BRCA1 and 2 gene-specific guidelines, but I'm not sure when this will be coming out. And I believe that's the end of my section. Thank you.
Thank you, Andrew, very much. And I think some of the sound was coming and going, but we heard most of it there. So thank you for doing that very much indeed. We're now going to move on to our second speaker, Miranda Durkee, who is going to give a whirlwind educational review of ACMG codes, how and when to use them. So Miranda, I'm going to hand over to you now, please.
Lovely. Thank you, Sandy. And thank you very much to Andrew as well for that great overview. Yeah, hopefully everyone could hear okay. And obviously this is all recorded, so you can go back to the slides as well. So the reason that we thought we would focus on the ACMG codes is actually from feedback from you from previous runs, that for those of us that are very familiar with the codes and start talking about PVS1, PM1, PS1, it can sound like a foreign language to those of you that are just learning what these codes are. So we thought we'd really take some time and go through each code and or the main ones that are very specific to cancer susceptibility, and some of the nuances or cautions of kind of when to use them and when not to use them. And I'm going to mainly concentrate on the CAMBIG guidelines, one because I'm very heavily involved in that, but also they're the most up to date. And as Andrew said, they are designed specifically for the cancer susceptibility genes. So just one final thing to say is that there are a few codes that I've included slides for, but we don't use them very often. And just for the sake of time, I don't want everyone to fall asleep. So I'm going to skip over a few slides, but yeah, you can always go back to this recording. So I hope everyone's got a strong coffee and we'll get stuck in. So I've grouped the codes into kind of areas into how you would use them. So the first kind of group is computational and predictive data. So this means that we don't have any experimental data for that variant, but we can use information about the type of variant or maybe some computational tools to predict what the effect on the protein is going to be. So I'm going to look at both the pathogenic and the benign criteria. So we're going to start with PVS1. And in the original Richards et al. Paper, PVS1 was the only code that you could use at a very strong strength. So it's a very, very useful code to use in order to get your variant to likely pathogenic or pathogenic. And it is to be used for null alleles. And by null, we mean where there's no protein produced. So you've got nonsense mediated decay, for example, a nonsense, a frame shift, a canonical splice variant, initiation codon, or a CNV. And yeah, obviously in a gene where loss of function is a known mechanism of disease, which it is for the majority of the cancer susceptibility genes. So as Andrew said, please use the PVS1 decision tree. It really is good. And also there's another kind of similar tree in the ACGS 2020 guidelines for the stop loss variants. So for the rest of this slide, I'm now going to focus on some of the complexities and when you either shouldn't use PVS1 or you should be very cautious about using PVS1 because this can really trip people up. So the first caution is that truncating variants in either the last exon of the gene or the penultimate 50 base pairs of the penultimate exon do not result in NMD. So you will get a protein translated and that protein may be functional. So a good example of this is in run 4, we included their classic BRCA2 benign variant, p.lysine 3326 stop. And for that variant, you really would not use PVS1 because we know that area of the gene is not conserved and that is a very kind of common variant in the population. On a very similar theme, also truncating variants in the first 100 base pairs of the gene may not be subject to nonsense mediated decay because you can get reinitiation from another in-frame methionine. So a good example of that, we've not included it in the scheme yet, but is RAD51C. You can get a p.lysine 4 nonsense mutation. And again, you've got an in-frame methionine at codon 10. So it would not be a good idea to use PVS1 for that variant. Then, as Andrew also mentioned, you can get frameshift variants at the end of the gene that cause a protein extension, which may not be subject to nonsense-mediated decay. So this example that Andrew already mentioned was in run 6 and caused a lot of problems in classification, and is already shown that using the PVS1 decision tree, we would score that as PVS1 moderate. However, you also need to check whether there is a termination codon in the 3' UTR, because if there isn't one, then you get a different mechanism called non-stop decay, and you therefore would get a null allele, and therefore you would apply PVS1 at very strong. So next, moving on to some splicing variants. And splicing variants can be very hard to predict. So you can get variants that lead to an in-frame deletion. So for example, this BRCA2 -2A to G variant causes a deletion of exon 12. That's an in-frame deletion, but more importantly, we know that that exon is functionally redundant in BRCA2 and therefore is not required for normal protein function, and therefore PVS1 would not be applicable in that case. So don't just apply PVS1 if you see a plus or minus 1 or 2. Then there are also splice variants where you can get an alternative in-frame cryptic splice site. So a good example is the BRCA1 C.301 plus 1 G to A. And this has been shown by RNA studies to cause a 3 amino acid deletion. So again, PVS1 really isn't the appropriate code to use here, and you'd be more wanting to use PM4, which is for in-frame insertions and deletions. Then this is a really interesting group, these splice variants where you get no alteration predicted, and these are all +2 T to C variants that can create a functional GC site. So in the ATM guidelines that Andrew's just mentioned, there's actually 6 of these variants that they've used Splice AI to check to see whether that splice site it does create is functional or not, and they've decided that PVS1 should not be applied in those cases. So yeah, really recommend Splice AI as a good tool for those. And then we also have CMVs, and CMVs you can think it's a large deletion, it's going to be pathogenic, but again they can create small in-frame deletions. So for example, in run 6 we had a BRCA2 exon 14 to 16 deletion. Using the PVS decision tree, that would come out as PVS1 moderate, but we were able to upgrade it to strong because the region was critical to protein function. So a good tip for that is to look for any known pathogenic missense variants in that region, and obviously please check that they're not acting on splicing. So that was a really kind of quick few examples of when PVS1 cannot be kind of so straightforward and you really need to take care in applying it. But there's a fantastic presentation on the CAMVIG website under the resources section that was done by James Drummond that's got absolutely loads more examples where PVS1 can create problems and certainly shouldn't be used at very strong strength. Okay, so next I'm going to move on to PM1 and PP2, which I've grouped together. PP2 are grayed out because it's for a gene constraint that we very, very rarely use in cancer susceptibility. PM1 is used where there's a regional constraint, and the CAMVIG guidelines, we would use it at moderate for these critically important residues in the ring, BRCT or DNA binding domain. And we would use PM1 supporting for other residues in those domains. I thought a good example to use for this code is that in run 6, we had a BRCA1 missense variant, p.glycine 1788 asparagine. And 30% of participants used both PM1 at moderate and PM5 moderate together. Anyone that's very eagle-eyed will have noticed that 1788 isn't in the list on the right of those key critical domains, so we would only use it as supporting. But also PM1 is saying it's a hotspot, and then PM5 is saying you have other pathogenic variants at that same codon. So they're kind of equivalent, they're saying the same thing, so you really would be over-counting that piece of evidence to use it twice, particularly 2 moderates. So CAMVIG guidelines, we don't allow these codes to be used together. On PM4 is the in-frame insertion deletions, and this is one of the ones I'm skipping over. Moving on to PM5, which I've just mentioned. So this is where you find a missense change, and there's been another pathogenic missense change reported at that same codon. And based on work that's been published by the CAMVIG group, looking at the Findlay assay, then we would use it at moderate if the variant you're looking at has a more deleterious Revel score than the previously reported pathogenic variant. However, we would use it at supporting if either you've got a less deleterious Revel score or it's been reported as lightly pathogenic in another patient, or only been reported in one individual. Basically, it's best to err on the side of caution because patients can have a very life-changing treatment on the basis of these results. So a good example from this is from run 4. We had a BRCA1 missense variant, p.threonine 1691 alanine. On the Findlay et al. Assay, a very, very good functional assay, there were 4 loss of function missense variants at this codon. There was 1 missense functional variant, and the variant that we asked you to analyze was actually intermediate on the assay. So we wouldn't use PS3 for this variant, but we could apply PM5. The Revel score was significantly was lower than the others, but above 0.7, which is our cutoff. So on to PP3, which is for the in silico tools or the multiple lines of computational evidence. And you've got 2 types of evidence you can apply here. The first is for the impact on the protein, and there have been quite a few publications showing now that use of multiple tools is no longer recommended because it can introduce significant bias. You can choose which programs you like or which ones you kind of want to make the answer that you want. So it's much better to use something like a really well validated meta predictor such as Revel, and we would use a cutoff of greater than 0.7. For splicing, as I mentioned before, Splice AI is a fantastic tool, but for anyone using Alima, as shown in the screenshot here, then using Maxent Scan or Splice site binder-like are also kind of recommended. And this is just showing an example from run 4, where this -3 C to G variant, pretty much you lose the natural acceptor site and you get creation of a cryptic site. Then I just really quickly wanted to highlight that there is a preprint now available from the ClinGen SVI looking at PP3 and BP4. And they have done lots of work that actually supports previous work that we've published via CAMVIG to show that some of the tools can be used at higher strength. So, for example, Revel can potentially be used at strong for PP3 and at very strong for BP4. So we have not incorporated this into guidelines yet, but watch this space, because I would say this is likely to be coming when this paper is published and out. So another code that can cause problems, and we've already received some questions about, is BP1. So this is a missense variant in a gene for which primarily truncating variants are known to cause disease. And there are 2 good uses of this. First, for genes where the vast majority of reported pathogenic mutations are truncating. So a good example is PALB2. Or the other usage is for gene cold spots. We often kind of talk about hot spots, but there are also cold spots of genes. So a good example of this would be missense variants in BRCA1 or BRCA2 that are outside of those key domains and they're not conserved. And by that, we just really want to check the conservation to ensure it's not highly conserved and it's not something that's to do with, a very key structural amino acid or something. But generally, it's fairly obvious when you can see it's not very well concerned outside of those domains, and you can apply BP1 at supporting. And a caveat, please remember to exclude splicing. So onto the other computational tools, which is BP4, the opposite PP3 for the multiple lines of computational evidence. And then we also have the BP7, which is for a synonymous or a silent change. And here you also have to look at the splicing, but you also have to check that the nucleotide is not highly conserved. So again, the same tools as before. A little caveat that some of the kind of older tools aren't so good at looking at the things like the GC donor splice site or the minor spliceosome, but Splice AI does work really well for those. And then also a note that in the ACGS guidelines, it was a few versions ago, I think maybe 2018, we allowed the use of both BP4 and BP7 together for synonymous changes so that you could get them to a class 2 rather than keeping them all at class 3. Okay, that was kind of a big section, but now we're on to functional data. So now it's just the PS3 and BS3. And the key thing here, so this is PS3 means you've got a well-established in vitro or in vivo functional study supporting a damaging effect. And as Andrew has already said, that we've got these fantastic Greenwich guidelines out. And before that, it was really hard to work out whether your assay was well established, was it good enough? So that really takes you through a framework of how to tell whether the assay meets the kind of minimum standards. And then the statistical analysis based on the numbers of true and false positives and negatives. We would recommend if the variant comes up as intermediate, you do not use the evidence at all because you don't know if that's intermediate due to the assay or intermediate due to the variant. And actually in cancer susceptibility, we're now very lucky that we have multiple of these very large multiplex assays. And so we need to consider how to use the evidence from different assays, particularly if it's conflicting. So just to highlight another useful tool on the CAMVIG website, in the top right-hand corner, and you click this button saying functional study scoring, it brings you up an XLS with all the main functional studies have all been analysed and assessed by the Greenwich guidelines for you, and it shows you what strength you can use, but all the kind of information and statistics, etc., is provided. And also in the guidelines, because this is from the consensus guidelines, we've got a couple of tables showing how you combine the evidence. So a good example of this is back in run 4, we had this p-dot isoleucine 68 to lysine in BRCA1, and that was found to be loss of function on both the Finley et al. Assay and the Starita et al. Assay. So we know that we can use PS3 at strong, but at that time, less than 22% of participants used PS3, and 70% of participants scored this variant as a VUS rather than a lightly pathogenic, which obviously has significant implications for that variant. So this run was before the Greenwich guidelines came out, so hopefully this is now improving as people are aware of them. So that the Greenwich covers really well the protein assays, but does not go into splicing at all. So there is a ClinGen SPI group looking at splicing, but there are no guidelines available as yet. So meanwhile, you just have to be very careful to make sure that the assay that you're looking at is well established and is a good quality assay. So some of the things to think about: has it been independently replicated? Have they used sufficient normal controls? If you detect an abnormal isoform, is that isoform naturally occurring? And is the splice effect leaky? So that means, do you get some wild type protein from that mutant allele? And so in the CAMVIG consensus guidelines, we have a grid showing you the strength that you can use, PS3 for the splicing assays, depending on all of those things. And a good example here, where we had 2 orthogonal splicing assays confirming out of frame transcripts. So we know we can be confident that we can apply PS3 at very strong. PS3, I'm going to skip over for the sake of time, opposite to PS3 really, and move on to population data. And I know this causes a lot of questions and can be quite a complex area. So hopefully you're going to find this helpful. So in the original Richards et al. Paper, the PS4 code was really designed to be used for case control studies where the prevalence of the variant in affected individuals was significantly increased compared to the prevalence in controls. But unfortunately, it's very rare to get case control studies in cancer susceptibility. So the majority of the published VSEPs have gone for this case counting approach, certainly used by the TP53 and the P10 VSEPs. And it's particularly useful where you've got quite a specific patient phenotype, and then they would not use that PP4 phenotype code. The advantage of it is that you don't need to know the total number of tests required. You don't need a big case control study. And so in the CAMVIG guidelines, we've said if there is insufficient data to perform case control analyses, then you can apply PS4 as supporting only. So being quite cautious because of how common breast cancer is in the population, but you can apply it as supporting if there are 5 or more observations of that variant in families with a family history of hereditary breast or ovarian cancer, and the variant is also rare or absent from NOMAD, i.e. you can apply PM2, which I'm going to come on to in a minute. So the other option, which works beautifully if you've got the data, is that we can actually do the case control approach by using kind of data that all the NHS labs now have been submitting data to NHS Digital, and NHS Digital have given that data back to the CAMVAR UK database, so anyone can access that data. We are due an update very soon, so we're hoping that the counts are going to pretty much double. And you can just use this very simple calculation called the Fisher's exact test to look at your cases with a breast or ovarian cancer, how many times you've seen that variant, how many were wild type, and then compare it to the controls, which we would use NOMAD for. So a quick worked example is this is the cases, and this is again from the CAMVAR UK website, we've seen this variant in the UK 18 times in over 22,000 total tests, and it's not seen at all in NOMAD. Before that we did this analysis, this variant was actually classified by most labs as a VUS. But you can see this data is very convincing. We get a very significant p-value, and we can use PS4 at a very strong level in order to push that from the VUS up to pathogenic. Okay, so now on to PM2, which is rare in controls, and it was originally intended to be absent from controls, but from the ESP and the 1,000 genomes project. So obviously we now have NOMAD and much bigger data sets. So as Andrew said, in 2020, the ClinGen SBI recommended that we downgraded PM2 to supporting only, but caveated that they anticipated that there would be adjustments in the relative weight of other evidence codes in order to accommodate that change. So the ACGS and CanBig together agreed that as we'd done a preliminary look at some of our variants, and that a significant number, particularly missense ones, would be affected and actually would move classification from likely pathogenic to VUS, that we would hold on for these other ACMG guidelines in order to balance this change. So the CanBig guidelines are quite stringent. We do allow the use of moderate, but only where there are 0 observations. So it has to be completely absent from NOMAD. And then we would use it as supporting where the variant is not absent, but it's a very low frequency. So we'd say 0.002%. And that's an example for PALB2. Oh, and yeah, just to say that for the primarily BRCA and ovarian cancer genes, we'd recommend looking at the female non-cancer cohort in NOMAD. So on to BA1 and BS1. So BA1 is the only code that you can use as a standalone to say it's really high in controls and that variant is obviously benign. Whereas BS1 is a strong code, just if that is too frequent in controls and then what you'd expect. And there is this fantastic tool that was developed by Nikki Whiffin et al. And it's located at CardioDB allele frequency app. And this can be used to calculate your maximum tolerated allele frequency for the variant in a control population, such as NOMAD. It can seem a little bit complicated, but the good news is that you don't need to do this because actually all that work has been done for you. So there, again, on the CanBig website under resources, there's a presentation on the application of the different thresholds for BA1, BS1, plus worked examples for the BRCA1, BRCA2, PALB2, and plus the Lynch genes. So I've included some of the calculations here. I'm not going to go through it, but we calculated that you could use BA1 standalone if the maximum tolerated allele frequency is greater than 0.1%, and for BS1 strong at 0.01%. But you don't want to kind of get that data directly from NOMAD because there is another calculation that looks at your Gaussian distribution. So you can either go to NOMAD and go to the top left-hand side where it says your pop max allele frequency, but that's just based on one population alone. Or you can go to the CardioDB website and go to this tab here that says calculate AC for allele count. So you put in your 0.01% and you put in your population from NOMAD, and this gives you your allele count. So a good example here is run 6. We had a BRCA1 C.301 plus 7 G to A variant with 13 alleles on NOMAD. So 38% of participants applied PM2 and 26% applied BS1. Hopefully, you can all now see that actually neither is applicable, that 13 is way too high to apply PM2, and yeah, we want more than 18 in order to apply BS1. So now moving on to the allelic data. Again, I'm going to go through this quite quickly because it's already been covered by Andrew. So it's not very often used, but there are some circumstances when you can use it. It's actually very useful because you can get up to kind of a reasonable strength, or we cap it as strong. So it's for recessive disorders where it's detected in trans. So this would be any of the genes that cause Fanconi's anemia, or you can also use it for ATM variants with ataxia telangiectasia. So we recommend using the SBI guidelines, but we would cap it at strong. With this caveat is that you need to be a little bit cautious about inferring the phenotype when it's just present in a heterozygous state, if the majority of the observations of it are seen in the biallelic or recessive state. So yeah, just be very careful with that. An example where we used PM3 was in run 7 for this PALB2 variant. This is another one I'm going to skip over and move on to the segregation data. And in actual fact, I didn't even do a slide for BS4 because it's pretty much never used, because it would be non-segregation of disease, but we know breast and ovarian cancer caused by BRCA1, BRCA2, HRR genes has reduced penetrance. You do see, and then we also have phenocopies as well. So it would be very unusual to use BS4 in this case, and it's a strong category as well. So we're just going to focus on PP1. So this is co-segregation of disease in multiple affected family members. We use the Jarvik and Browning approach, which is kind of shown here, the figures that you need. And in these circumstances, you would only count the affected relatives with the variant. So if you had an intervening relative who had the variant, but who hadn't developed breast cancer, you would not count that relative. And although you think it would be very useful in an autosomal dominant disease, actually it can be quite hard to apply because you need at least 3 informative meioses in one family, each of them with the variant confirmed. And because cancer can be a later onset, it can be tricky. So a good example to demonstrate this, we had in run 6, this BRCA1 splice variant. There was this fantastic paper by Garibe et al. With all the beautiful pedigrees, which is very rare that you see this. But actually, when we looked closely at each of these pedigrees, 3 out of the 4 were not informative for that segregation to affected that had the variant. So we could only use one family, and we were only able to apply PP1 at supporting. Okay, so my last grouping, which is the multifactorial data, and this is something that's quite new to the CAMDIG guidelines. So this is something that we implemented in the BRCA1 and BRCA2 gene-specific guidelines in June last year. And it's a way of using the published multifactorial data from Enigma. So while we're pending the Enigma guidelines being ratified by the ACMG, and how we could kind of translate their results into the ACMG codes. So the shown here in the text is how you would say, for example, if you've got a very high likelihood ratio based on segregation, you could apply that under PP1, or if you had a very low negative score, you could apply it under BS4, or specificity familial tumor phenotype, apply under PP4, etc. So, but sometimes where there's evidence in several of the categories and each of them individually doesn't really add up to kind of significant points, then you can use the totality of the evidence, and we would recommend applying this within PP5. So obviously I'm aware within the ClinGen SBI group, they got rid of the use of PP5 and BP6, the reputable source, because they recommended that you went and got that data specifically. But I think for this circumstance, this is a really good use of PP5 because you're combining different codes. There are multiple of these multifactorial assays published, and if your variant is in more than one, then we would recommend that you use the most recent publication. And a good example of this is in the run 6, and we have this BRCA1 C.301 plus 7G to A variant. It was an Enigma class 1, so lots of participants classified it as class 1, but using the ACMG, we could only get it to class 2, likely benign. So this is pre the CAMDIG guidelines coming out in June 2021. and that we use BS3 strong, and we did still use the BP6, but just at the Richards et al. Kind of default strength of supporting. But now you can convert the likelihood ratio, and you've got several of these codes that are very high. So, and the totality of the evidence combined likelihood ratio is -32. So you can really see that you've got a huge amount of evidence and that you are able to use BP6 at strong and then get that to class 1. And then my final 2 slides is the other area we get a lot of questions about and can be a very tricky area, is how you combine the codes and which codes you can and can't use together. And Dr. Alice Garrett from the Canvig group has produced this really useful grid that's in the consensus specification, really mapping out each of the codes. So where it's shown in light green is you can use those codes together. Where it's in the light green, the darker green, sorry, with the number, you can use them, but there is a caveat that's shown in the table on the right, and those in red are not permitted to be used together. And again, that's all explained on the grid on the right. And we have exactly the same for the benign codes. So hopefully you'll find this useful when you're classifying your variants. And yeah, as Sandy said, please do provide us with some feedback. And that's it from me. I'm gonna hand you back to you, Sandy. Straight to Kathleen.
Ida, Miranda, that was fantastic. Thank you very much. It's so helpful to see things broken down into little chunks, because it's quite overwhelming when you just look at the guidelines and don't really know where to start. So that's great. Thank you so much. And then moving on, Kathleen, can we come to you now for to try and describe how we use our databases and all the multiple databases that are available for us as tools? That would be great.
Sure.
Great.
Yes.
So next slide, please. We'll move on.
So as Miranda and Sandy already told you, I will continue, because one of the recurrent questions we received on the previous runs was about databases. Which databases can we use and to deal with them? And this is coming from the criteria PP5 and BP6, where it is said that you can consult reputable sources and to use as a criterion to as pathogenic at a supporting level, or at benign as a strong of supporting level. But what is a reputable source? Miranda already hinted towards the likelihood ratios that have been calculated by Enigma, specifically that for BRCA1 and 2 variants. However, since we have to evaluate variants also outside BRCA1 and 2, we need some more guidance. And ACGS has detailed this in more detail and have indicated that we can use the criteria, the classifications done by large laboratories who may have large series available of cases that are not publicly available, but which allow them to reliably classify the variants. So they state that you can use the classifications done by 2 or more accredited North American commercial diagnostic laboratories, if they have done it after 2018, or if there is one North American commercial diagnostic laboratory where they have cited that has been cited in the literature, or when there is a ClinGen expert group who has classified the variant. So where to find this information? Well, the best known database, of course, is ClinVar. It's an active partner of the ClinGen project. So they archive all the results of the recognized expert panels and also of the providers of best practice guideline. However, you should know that ClinVar does not curate the content and does not modify interpretations independent of an explicit submission. So you have to be careful, and sometimes you may have evidence available that the classification in ClinVar is not correct, and ClinVar then motivates you to submit your own data with the evidence supporting your interpretation. To give you an idea about the reliability of the classification in ClinVar, they use a four-star rating system. So if a variant has only 1 star, this means that there is only a single submitter who has submitted this variant and who provided criteria. So for instance, you have here a variant from run 7 that was submitted by 1 lab. Yeah, you cannot, and there were contradictive. There were several labs, there were 5 submissions, however, but there were contradictive interpretations, meaning there were 3 labs classifying it as uncertain significance and 1 as benign. So therefore, you cannot see it as a reputable source, and you cannot apply PP5 or BP6. You can have stars in ClinVar, and this means that there were criteria provided by the submitters. There were multiple submitters, but there were no conflicts. And the more reliable is when they are coming from an expert panel, because then you will see the variant will get 3 stars, and then there is an expert panel who has done the assertion panel. It's like Enigma, or for the Lynch genes, it can be Insight, and this can be then copied. And for instance, in case of this variant, it's a silent variant, was classified by Enigma, you can apply BP6. There are also 4 stars. However, this is then based on practice guideline assertions, but there are no examples for variants in the BRCA and HRR genes, so I will not further discuss this for the time constraints. Besides ClinVar, there is LOVD, and LOVD is the Leiden open source variation database, and it's a software that helps to collect genomic variants and phenotypes. And the big difference with ClinVar is that it's curated. It allows you to see both a gene-centered view, but also patient-centered view. So if you want to have an overview of all variants in a particular patient that was submitted, you can also see this. LOVD is open source, and the designers try to actively further improve it, and it connects with data resources like GNC, CBI, ABI, and Utilizer to make sure that the variant nomenclature is correct. There are several databases available for the genes that are relevant for this audience. So you have for BRCA1, BRCA2, ATM, PALB2, CHECK2, and you can easily see who are the curators. These can be contacted. However, for some databases, there is not yet a curator appointed, and there is a vacancy. Showing you the variant that I have previously shown for ClinVar. This is how it looks like in LOVD. So you have this missense variant in ATM. You see how the lab has or the submitter has classified it, and in some cases you get some phenotype information. For the others, use it's a sharing initiative from the Netherlands, or you can have labs who put their classification criteria so that you can do it, judge further for yourself. However, so you have ClinVar, you have AlloVD. There are also initiatives who are trying to bundle all the information from the different databases, for instance, BRCA Exchange. So this is a huge effort to do it for the BRCA1 and BRCA2 genes, and you see that currently the database counts more than 68,000 variants in any of these two genes, but it's limited to these two genes, so it's only available for BRCA1 and 2 and not for the other genes. They bundle information from multiple databases, which gives you immediately a good overview. I will show in some examples in the next slide. And they aim to provide the researchers with a set of BRCA variations and annotations, which is as comprehensive as possible. And that's an example you can see here. So it's a variant Miranda has also discussed. It's a splice variant in BRCA2. You immediately get an overview of the frequencies in, for instance, GNOMAT in the different subpopulations. You immediately get an overview of how it has been classified in ClinVar and who did this, and also who has submitted it to AlloVD and their classification. What I particularly like about the BRCA Exchange is that they also give an overview of the functional assay results that have been performed. So here for another variant Miranda has discussed in BRCA1. You get an overview of the functional assays that have been evaluated by Enigma and that are considered as very well validated. And you immediately get an overview if the functional assay is based, evaluates the variant at the RNA level or at the protein level, or at both levels. So for instance, you have here the Findlay assay, where you immediately see that this variant was identified as a loss of function variant. If you click on this, you get this overview and you have some more detailed information. Miranda already hinted towards the work that has been done by CAMVAR UK, and this is actually bundling also a lot of providing an overview of a lot of information. And the big advantage of this database is that it's broader than only BRCA1 and 2. So for instance, I'm showing here the results from the same variant as I have shown for the BRCA Exchange data. It looks a bit different. Here you have 6 tabs on which you can click and have some information. For instance, in the summary tab, you get this overview. Then for the controls and the frequency, you get these data from the GNOMAT population frequencies. You have for the genetic epidemiology, you get the likelihood rate issues as published by Parsons et al. And there is also a functional and splicing assay, and that is these results are shown here. There is a 6th step. This is only accessible for people with a login, so having an NIH affiliation, and for me this is not the case, but all the other information is available, except this one is limited to people who are associated to NIH. Here in the overview, they also hint towards 2 other databases, which I have not yet discussed, is the HGMD and UMD classifications. What are these? Well, the UMD BRCA1/2 mutation database was initially established by the French academic laboratories, and it was well, freely accessible until 2015. But since then, they have well, they have more commercialized it, and access is behind. Well, you have to sign in for a license. So this is, well, this is less accessible today, and the same counts for HGMD. So until 2015, it was freely accessible, and it attempts to collate all the known published gene lesions responsible for human inherited disease. So this was not coming from one or a group of labs. This was really published data. But since it's now you also need a commercial license to get access to the HMD professional database. So I would like to end here and hand over the microphone back to Sandy and Simon for the Q&A session.
Fantastic. Thank you, Kathleen, very much indeed. What a great compliment, the three webinars together. The presentations have really been a very comprehensive overview and huge educational value. So thank you very much indeed. Next slide, please. So just before we move on to the Q&A session and I hand over to Simon, can we just move on to the next slide, please? I just want to flag that currently the BRCA1, BRCA2 and HRR gene variant Run 8 is now open. It's available until the end of this month, that's the 30th of June. So if you've not yet registered for the online individual assessment, then please do so. And here is the link. We will be able to hold a webinar in the middle of July following up from the results and sort of the expert panel giving you feedback on the variants in this EQA run. So really please do participate to get as much benefit from your learning today as well. If you have any problems or want some further information, then please contact either the EMQN office or the GenQA office. Thank you very much indeed. So Simon, over to you for a Q&A. I see we've got quite a stream of questions coming in and I'm sure we can cover a few in the next few minutes.
Thank you, Sandy, very much indeed. And thank you to Andrew, Miranda and Kathleen as well. We've had quite a few questions coming in already, which I can see that the team have actually been answering as we go through. Given the timing, we haven't got time for very many, but we've got a couple of questions here that we've queued up for you already. So we're going to kick off for a question for Miranda, which is, do you consider Finley et al. As a standalone assay? And if you do, can you use PS3 in this case if you only have Finley et al. Data?
Thank you. So one of the principles of the ACMG guidelines was that you always had to have two pieces of evidence. So the only standalone code you can there is the BA1. So you would need a second piece of evidence, and I think this is especially important when you've got this functional data because they did such a big multiplex assay using the saturation genome editing for every possible position. There are lots of those variants that have actually never been seen in a cancer case. So we've got the functional data, but we don't really have the data from real people to know that it causes cancer. So I think it's really important that we have another criteria you use.
Fantastic, thank you, Miranda. So while you're on this question stream of questions coming in, I've got one more for you.
Okay.
Do you use co-segregation PP1 or the intrans PM3 criteria when the variant has been identified in a patient with other phenotypes different from cancer?
Very, very cautiously, I would say. So yeah, we know that there are hypermorphic variants that can be present in patients with Fanconi's, and so I certainly wouldn't use PP1. PP1 has to be for a cancer phenotype. I think you probably would use PM3 cautiously if there are kind of other cases or other evidence. If that was the only case, and say you were testing an unaffected person, I would be extremely cautious about using that.
Okay, so we've probably got time for one, maybe two more questions, and I can see that we have 2 questions to go. So that's very handy. That wasn't scripted, I can assure you. So can we use RNA sequencing analysis to reclassify variants? So if an intronic variant causes a splice alteration, can I use PVS1 decision trees? Anybody like to take that question up?
That's a very good question. I do think we're waiting on some guidance from the, I believe there's a ClinGen working group looking at RNA. Oh, I apologize, I forgot to turn on the video. I believe there is a ClinGen working group looking at RNA-seq or RNA and splicing. At the moment, I would probably feel most comfortable using this as functional evidence. But that's
Yeah, I think some of the research are starting to use PVS1_O for observed, but again, that's coming with the next version of the ACMG. So at the moment, we're still using it under PS3. I think that will change.
Okay, so there's definitely guidance coming on that very shortly, is what you're trying to say, and it's certainly going to be incorporated into classification at some stage in the near future.
I don't know how near, but
well, sometime,
I don't think we'll get the ACMG new guidelines this year, but maybe the splicing SBI. Okay,
right. So we finish off with one last question, throwing open to the audience, to the panel as well. So PP5. This is quite a complex question, but greater than 2 accredited North American US laboratories classify Class 4, Class 5 applies with 2 stars, but the literature cited in ClinVar by the submitters refers to a similar variant. The question is, can PP5 be used for this variant in question?
Maybe I can take that. Well, it says, the question is, it's a similar variant. So then I would rather check if you cannot apply the PS1, because this is about a variant in the same amino acid, and then a bit dependent on if it's really leading to the same amino acid change, you can check if you can use PS1 or not. And if if PS3 about the functional assay can be applied.
Okay, thank you, Kathleen. Right, so that probably brings it to a conclusion for the questions and answers. We have all the questions available to us on our Q&A, so we will collate that information for you and make it available. I suspect probably on the scheme report after the assessment process is concluded. As Sandy quite outlined, the actual variant assessment run is still open and will close towards the end of the month. So if you have not registered, please make sure you do so as soon as you can, so you can join us in that assessment process. We have another webinar, as I said, middle of July to feedback on it. So it only leaves me to thank our expert panel, to Kathleen, Miranda and Andrew for their amazing talk. It was a really good and hopefully very informative webinar for you guys. If you have any questions, please do not hesitate to get back to us via the AZ team or the EMQN or GenQA offices. I'm sure you all have the email address. And with that, I think we'd like to wrap up and conclude. And thank you once again for joining us for our webinar today. Thank you very much indeed, and good afternoon.